Imagine a single molecule controlling hundreds of genetic programs simultaneously. MicroRNAs are endogenous, single-stranded, non-coding RNA molecules typically ranging from 19 to 25 nucleotides in length. These molecules function as key regulators of gene expression, operating primarily at the post-transcriptional level. MicroRNAs bind to specific target messenger RNA molecules, usually within the 3' untranslated region. This binding is often characterized by incomplete complementarity. The result of this imperfect match is the repression of protein translation or the acceleration of messenger RNA degradation. A single microRNA molecule possesses the capacity to regulate hundreds of distinct target messenger RNAs, establishing them as powerful master switches within cellular control networks. The discovery of microRNAs fundamentally demonstrated that a vast portion of the genome is dedicated to regulatory non-coding elements, changing the understanding of genetic information flow. MicroRNA biogenesis initiates in the nucleus, where microRNA genes are transcribed by RNA polymerase II into long primary transcripts called primary microRNA. The primary microRNA transcript immediately folds into a characteristic hairpin structure. The nuclear microprocessor complex, which consists of the RNase 3 enzyme DROSHA and its cofactor DGCR8, recognizes and cleaves this primary microRNA. This cleavage releases a shorter, approximately 70 nucleotide precursor microRNA. The precursor microRNA hairpin is actively transported from the nucleus into the cytoplasm by the nuclear export receptor exportin 5. Once in the cytoplasm, the precursor microRNA hairpin undergoes further processing by the RNase 3 enzyme dicer. Dicer excises the loop structure from the hairpin, yielding a double-stranded microRNA duplex, which is typically 22 base pairs long. This precise sequential processing ensures the correct size and structure for regulatory function. The resulting double-stranded microRNA duplex is immediately loaded into the RNA-induced silencing complex known as RISC. During this loading process, the duplex is unwound. One strand, designated the guide strand, is retained within the RISC complex, while the complementary passenger strand is typically degraded. Argonaute proteins, specifically Argonaute 2, form the catalytic core of the RISC machinery. The retained guide strand directs the RISC complex to specific target messenger RNAs based on sequence complementarity. This targeting specificity is primarily dictated by the seed region, which comprises nucleotides 2 through 8 of the microRNA. If the complementarity between the guide strand and the target messenger RNA is near perfect, Argonaute 2 acts as an endonuclease, resulting in the direct cleavage and destruction of the target messenger RNA. If the complementarity is imperfect, the primary outcome shifts to translational repression, often followed by the sequestration of the messenger RNA into cytoplasmic processing bodies for eventual decay. MicroRNAs are essential regulators governing fundamental biological processes, including cellular differentiation, proliferation control, programmed cell death, and metabolic homeostasis. Dysregulation of microRNA expression is a defining characteristic of numerous pathologies. Specific microRNAs function either as tumor suppressors, such as microRNA 34, which targets cell cycle genes, or as oncogenes, termed oncomiRs, like microRNA 21, which promotes proliferation. These regulatory roles are frequently altered in cancer. For example, the deletion of the microRNA 15A and 16-1 cluster, which normally targets the anti-apoptotic protein BCL2, is strongly associated with chronic lymphocytic leukemia. Changes in microRNA profiles are not merely reflective of disease progression, they are often causative factors. Altered microRNA signatures are also observed in cardiovascular, neurodegenerative, and autoimmune disorders, correlating directly with disease severity. Circulating microRNAs exhibit remarkable stability within biofluids such as serum, plasma, and urine. This stability is achieved because they are protected from RNA degradation by encapsulation within extracellular vesicles, specifically exosomes, or by binding to high-density lipoproteins or argonaute proteins. This inherent stability makes them ideal non-invasive biomarkers for liquid biopsy applications. Quantitative polymerase chain reaction and next-generation sequencing are the standard methods utilized to profile these circulating microRNA signatures. Specific panels of microRNAs can be employed to distinguish early-stage cancers from benign conditions, monitor the efficacy of therapeutic interventions, or predict the rejection of organ transplants. In clinical microbiology, analyzing certain microRNA ratios offers rapid diagnostic potential by differentiating accurately between viral and bacterial infections. MicroRNA-based therapeutics pursue two main strategies: inhibiting pathogenic microRNAs or restoring deficient ones. Anti-miRs, also known as antagomirs, are synthetic, chemically modified oligonucleotides designed to bind with high affinity and specificity to target microRNAs. This binding effectively sequesters the microRNA, preventing its loading into the RNA-induced silencing complex. This inhibition strategy is applied to treat diseases driven by the overexpression of oncogenic microRNAs. Conversely, microRNA mimics are synthetic double-stranded RNAs used to replace tumor-suppressive microRNAs that have become downregulated during disease progression. Delivery remains the primary technical challenge for both therapeutic approaches. Advanced formulation technologies, such as lipid nanoparticles or targeted conjugation, are required to ensure efficient uptake by the intended tissue or cell type while simultaneously minimizing undesirable off-target effects throughout the body. The mechanism of microRNA action differs significantly in plants compared to animal systems. Plant microRNAs typically exhibit near-perfect complementarity to their target messenger RNAs. This high degree of matching results almost exclusively in argonaute-mediated cleavage and subsequent degradation of the target transcript. Plant microRNAs are crucial for regulating developmental timing, establishing organ morphology, and mediating responses to environmental stress, such as nutrient deprivation. In microbiology, host microRNAs participate in antiviral defense by directly targeting viral transcripts, exemplified by microRNA 122 targeting the hepatitis C virus genome. Conversely, many viruses, particularly DNA viruses, encode their own viral microRNAs specifically to manipulate host gene expression, often suppressing immune responses or promoting viral latency.